In part 2 of the search for genetic material, we ended in 1952 with Hershey and Chase, who proved that DNA and not proteins are the genetic material for organisms. And in 1953, Watson Crick and Rosalind Franklin had another major breakthrough through something called X-ray diffraction. They discovered that DNA was a helical shape with two strands. In the same year, Meselson and Stahl used the double helical model to also determine the method of DNA replication. So at the time, scientists thought there were three main possibilities. Conservative replication assumed that the old strand of DNA would stay intact within one of the daughter strands, and the second daughter strand would be made of completely new nucleotide bases. In the dispersive replication model, it assumed that the two daughter strands would have a mix of old and new DNA, sort of dispersed amongst the entire DNA strand. And in semi-conservative replication, the model assumed that the daughter strands would have one old strand of DNA and one new strand of DNA. And so Meselson and Stahl performed an experiment that went something like this. They first grew bacteria with nucleotides that had a heavy isotope of nitrogen, and we represent this through this pinkish color. And they grew the bacteria within these isotopes because the bacteria would then be able to incorporate the heavier nitrogen into their genome, into their DNA. After several generations, they switched the bacteria into a different solution of nucleotides with lighter nitrogen isotopes, and the light nitrogen we represent through this sort of fluorescent yellow color. And so the reasoning behind this, they would now be able to differentiate between the old DNA and the new DNA, because the old DNA would have heavy nitrogen, whereas the new DNA would have light nitrogen. And if you put these DNA strands within a tube, you can easily distinguish because they're different weights. So the heavy nitrogen would sink lower down into the tube, whereas the light nitrogen would sort of float higher on top. And if the conservative method were true, after the first generation of DNA replication, the tube would have two bands of DNA, one for the heavy strand and one for the light strand. Whereas if the dispersive model were true, then there would only be one band in the middle that sort of averaged out the weights, because both daughter strands would have both the light and the heavy nitrogen. And same for the semi-conservative, half of the strands were heavy, half the strands were light, so the weight is averaged out in this one single band in the middle. So what Meselson and Stahl actually observed, they observed only one band. That means conservative replication cannot be true. So how did they distinguish between whether it was dispersive or semi-conservative replication? They had to allow the bacteria to replicate their DNA in a second generation, so it would look something like this. And if the dispersive model were true, then there would only be one band of DNA within the two that sort of averaged out this band and the light nitrogen band. And the logic behind that, it's there are about 2/3 of the DNA is made from the new light nitrogen isotope, and 1/3 is made from the heavy nitrogen isotope, or it might be 3/4 and 1/4. Either way, the band would be closer to the light nitrogen isotope, but and farther from the heavy nitrogen isotope band. So if semi-conservative replication were true, then there would actually be two bands. There would be one band for light nitrogen, to represent these two strands right here, because they were all made of only light nitrogen, and there would be another band, again, with a averaging of the light and heavy nitrogen isotopes, because it would have half heavy nitrogen, half light nitrogen. And what they actually observed in the second generation of DNA replication was this double band in the tube. And so this confirmed that semi-conservative replication is the model of DNA replication. So these are the 5 major milestones that are important to know, not in great detail, but have a general sense of the timeline and the major experiments we mentioned: Griffith and his mice, Hershey and Chase with their bacteriophages, and Meselson and Stahl with their weighted nitrogen.